Today in lab, you're going to go through looking at roots. So I'm going to spend the first bit of lecture here pointing out some key things now about roots. And then when you walk into lab today, you'll get it a second time, and that will help you learn the material. And then the rest of today's lecture, I'll shift back into covering photosynthesis, okay? I'm still reviewing the lecture exam questions. I go through each question and I look at the statistics for each question. And if I see something weird going on, I adjust points. So I have yet to do that. But if you want to go through your lab exam in more detail, please set up an appointment with me. I'd be happy to do that. Go through it. We can talk about strategies if you're wanting to improve your chances to do better on the next exam. Certainly willing to help you. You know, it's a lot of jargon, just like with any field of study, anyone. It's kind of like learning a foreign language. So there are some ways to do that more efficiently and easily when you go to study that, right? Also, I will be posting an assignment, a take-home assignment, essentially homework. It'll be in EduCat, and it'll look like, you know, the icon that shows a quiz icon, but it's not really a quiz, it's a take-home assignment. So I'll be posting that into EduCat shortly. It's with regard to photosynthesis. I want you to use that as a way to help you focus in and learning the key concept, and that you work through it, and it's all open book, open note. You just work through it and try to answer it all correctly. You'll get all the points for it, right? So I will be posting that, and then I'll send out an announcement when it's ready. Usually when I do these kinds of assignments, I'll post it into EduCat, alert you about it, and then you typically have a week to get it done, okay? So obviously I want you to have it done before the next exam, and I need to have it done in advance enough so that I can look at the assignment and catch any misconceptions and help you get that corrected. Okay, so just keep that in mind. Look for an announcement from me on that. But let me talk to you a little bit right now about roots. And so today's lab, you're going to be looking at them. You're going to be doing a little fun exercise with them and with materials that we can extract from roots. But basically, I love this image. Whoever took it was on their back looking up into the plant canopy. Imagine, you know, this is kind of the view the roots would have if they could look up. So roots are incredibly important for the plant for a variety of reasons, obviously. And a lot of artists, when they draw the root, they tend to do an extreme of, like, this is a taproot idea, where you have that central major thick root and then smaller lateral roots coming off of it. And that is certainly a design of roots, but roots can have a variety of different designs. And here you can see some of their key job duties in terms of what are they doing for the plant. So you'll be looking at some storage starches, for example. You'll be looking at that today in lab, as well as looking at root anatomy. But when you look at root systems, you need to keep in mind that there's all different variations. You have the one extreme of a taproot system, and then you have the other extreme that's called the fibrous root system, where there is no central taproot. All the roots pretty much look the same, all right? And then there's things in between this. So it just depends on the plant species and what habitat they've adapted to. And of course, roots can be very, very deep into the soil, or they can be much more shallow and only be in the surface regions of the soil. And again, that's driven by what is the habitat they're adapted to, what's the quality of that soil. And so you have a lot of variation. When you're looking at roots, I do want you to know the anatomy here that they're showing in longitudinal sections. So if you cut the root lengthwise and look at it under the microscope, you would see something like this. And so the very tip of the root area, of course, you have the root cap region, which is epidermal cells that are just built up a little bit to help protect the root tip. So as you know, soil is pretty abrasive, and so you need to have a little bit of protection as the root is growing through the soil. And then right behind that, you see the zone, region of cell division, or zone of cell division. So that's where you have the apical meristem. tissue, those precious cells that are going to be constantly dividing and growing and leading to the length of the root. And so as the root is developing, you'll also see this region called the elongation region or zone of elongation. That's just where you start to see the cells getting bigger. They get bigger, they reach more of the mature size. And then beyond that, as you go up the root, you'll see a zone of maturation. That's where the cells have finally gotten to their, length and size that they should be. They have all their internal organelles up and running. Everything's going to plan. And then that's also where you see root hairs. Root hairs showing up is in the zone of maturation. So you tend not to see any root hairs further down toward the tip. So you'll look at some radical root tissue today under the dissecting scope. And then this right image is just looking at a really young root that hasn't quite grown up yet, where you have a lot of pretty even looking cells. All this yellow region is, eventually that's going to become mostly parenchyma cells. And then this red region, that's going to become the vascular tissue, the xylem and phloem. So there's a lot to this. When that seed is germinating and growing, the root tissue is undefined initially, and then it will grow up into what you see more like in figure A. All right, so there's a lot of growth and differentiation that needs to take place to get a really intact, healthy, well-functioning root. So if you take a root and cut it in cross-section and look at it under the microscope, you would see something like this. And so this is an example of a dicot root. And a dicot root tends to have, in the very center there, you see kind of that plus shape or cross shape of big red cells. Those are the xylem. And a dicot root tends to have that pattern where you'll see that cross or plus shape of the xylem. And then in each crook of the plus mark, clusters of phloem are sitting there. Okay, so the big red, remember red, when they stain plant tissue, they usually use fluoroglucinol. That will stain lignin red. All right, so anytime you see red, think about a secondary cell wall. In this case, it's xylem cells that we're looking at. So a dicot root, you'll see something like this. The other thing to take note of when you're training your eye, because you're going to be looking at roots and stems this week, and you need to tell the difference. What's a root versus what's a stem? And the way to do that in cross-section like this is you look at the placement of the vascular tissue. Where is it with respect to everything else going on in that cross-section? If it's in the dead center like that, it's a root. All right, so that makes sense to put your vascular tissue into the center. Think about roots buried in the soil. There's all kinds of critters coming along that could potentially chew on that root. Makes sense to put your vascular tissue that's so precious, where that's how you get things sent through your whole body. Stick it in the middle where you have some protection, so that when critters are coming by in the soil, they can't easily get to that. Because if they damage the vascular tissue, you're going to have a much tougher time. So stick it in the middle like that. And then, as you can see, with the laser pointer, it's kind of right where the laser pointer is kind of in between the xylem there. So in each corner, so to speak, of that plus mark, that's where you see the xylem clustered. So the clustering is in 4 regions there of the phloem. Right, so that's one thing. And then as you blow that up a little bit, so this right image here, they've blown up the vascular tissue. So again, you can see the red xylem and then the kind of bluish green phloem in there as well. And then all around that vascular tissue is this ring of red cells. All right, that's the endodermis. That's another thing you should train your eye to look for. And so the endodermis is that ring of red cells. It's got some lignin in there again, all right, top red. And then the other thing to pay attention to, right to the inside of the endodermis, is another ring of cells, more delicate looking, called the pericycle. Those are living cells. and they're a region of active growth. So you can think of them like meristematic tissue again. The pericycle cells will routinely divide if that root needs a lateral root coming out. And I'll show you some pictures of that. Okay, so that endodermis kind of surrounds and protects the vascular tissue, and then just inside of the endodermis is the pericycle. I'll get into more of this when we start getting into roots for real in lecture, just giving you some visuals to look at and think about here before you walk into lab. So here's this idea of a lateral root. So you can actually, the way the artist has drawn it, this thing coming off here, off to the left, that's going to be a lateral root. So it originates in the pericycle region. Those cells divide, and it allows for this outgrowth of a lateral root. All right. The bulk of the root tissue, all these other cells that you're looking at, those are just parenchyma cells. There's a bunch of parenchyma cells. And then, like you can see this one, and then let me show you the next one. You see all the purple dots in there in the parenchyma cells? That's a bunch of starch. Those are amyloplasts. So roots are really good at storing starch. All right, and so you can see this root is just chock full of amyloplasts. All that purple is picking up on the starch inside the amyloplast. And then again, you can see the vascular tissue in the dead center of the cross section here of the root. Another thing you'll look at today is the difference in anatomy between a monocot root and a dicot root. So we're zooming into the angiosperm, the flowering plants, and we're thinking about monocot versus dicots. So a dicot root has that plus shape, the di meaning two. I like to think of it as two parts of the xylem forming that plus shape right in the center. All right, and then if you look over on the right, this is looking at a monocot root, where you have a single ring of vascular tissue. Instead of in the plus shape, it's a ring of vascular tissue. So very different design between a monocot and a dicot. So please start training your eye today in lab to that as well. How do they design their vascular tissue setup? So let's look at that a little bit more carefully. Here's the monocot root, where you can see that ring. So let me enlarge this a little bit. So again, the xylem, like all those big cells that you see in the center, the big fat ones, those are vessels. That's part of the xylem. All right, so vessels are really wide in diameter like that. And then the smaller kind of purpley red cells in there as well, those are the tracheids. So here we can see, remember, angiosperms can have vessels and tracheids as part of their xylem. Here's an even better image of it here, where you can see that ring of vascular tissue in the monocot. All right, and so all those big whopping cells, huge diameter, those are vessels. That's part of their xylem. And then the smaller ones around that, the reddish purpley ones, those are tracheids. Still talking about xylem. And then all the more delicate kind of blue cells that are to the outside there, that is the phloem tissue. All right, so please start training your eye to looking at what's xylem, what's phloem, how does it look when you're looking at a root, monocot versus dicot. And then these things shooting off, those are the lateral roots that are starting to grow and come out. So you'll be looking at that in lab today, okay? And so that's about it, I think, that I wanted to clue you into for the root section. Are there any questions on any of the root anatomy right now? So as you're studying in lab, make sure that you're really paying attention to what's going on with the monocot root versus the dicot root, what's similar, what's different, and start training your eye to that. And then on Thursday, you'll look at stem tissue. So in Thursday's lecture, I'll also give you a little bit of info on the stem tissue. And then again, monocot versus dicot. We have to think about that. Okay. So roots are amazing part of the plant, and you'll have some fun with them today in lab. all kinds of very dynamic interactions going on when you look at roots buried in the soil and all the organisms that interact with the root tissue. It's really a incredibly dynamic part of the plant. And what most people don't realize is that the root biomass of the plant is typically much higher than the biomass above ground. So the same plant, it actually has more weight biomass in its roots compared to its shoots. And so we don't really appreciate that, of course, because we never really see root systems that are buried in the soil. But they can be massive and much more biomass than what you see above ground. Okay, so let's shift back into the photosynthesis PowerPoints. And so we're looking at the idea of the light reactions we've talked about. And again, very big picture idea. I'm not going into detail on that. And then here you're seeing how it links into the Calvin cycle. So when all is said and done, with the light reactions, there's two really important molecules, right? You got the ATP and the NADPH. And so when those are being created through the light reaction processes, that's really good. And they get made in the stroma of the chloroplast, and they can immediately start feeding into where they're needed to get the Calvin cycle to run. So we've been focusing on the Calvin cycle and working through that. And so here, we looked at this last time, where we're thinking about this cycle where you start with RUBP and CO2. And so the enzyme that's facilitating that is Rubisco. So please make sure you're keeping these acronyms straight. Rubisco is the enzyme. RUBP is the 5-carbon molecule that the CO2 is going to react with. All right, so Rubisco will take in CO2 and RUBP to its active site and react those two. And if everything goes okay, you're going to end up with 2 identical molecules of PGA. But that all happens spontaneously. So as long as Rubisco is surrounded by CO2 and RUBP, this will just happen. There's no input of energy, no ATP, no NADPH. It'll just happen. Now, once the PGA is formed, you see, okay, now ATP has to come into play here. You have to use that to do this part where we go from PGA, and then that will get, excuse me, phosphorylated, so that you, and then several steps here, and then you end up with G3P. So the second stage of the Calvin cycle is taking that PGA and working it to G3P. Now the cycle is happening over and over all day long. And so what happens is you can start to get a buildup of this G3P molecule, which is really good because that's the point where that G3P can get sent off into another pathway to actually make sucrose that the plant needs. Okay, so that's why you see that arrow green box there. They're saying, let's take some of that G3P and send it into a pathway so we can make sucrose. So that's great. And if everything's going okay, you get this, we call it a pool of G3P molecules that get cycled off into the pathway for sugar, for sucrose synthesis. But then you need to make sure that this Calvin cycle keeps going all day long. So you have to make sure that you make more RUBP so that CO2 is coming in and has something to react with. So that's where you see this last phase of the Calvin cycle is that G3P, and then they go arrow, 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 and then you end up with ribulose 5-phosphate. So we're close, almost there. But you need one more phosphate tacked on. So again, coming into play. So the ATP is needed at that key step as well in order to generate fat. So the first part of it, when RUBP and CO2 are reacting, no ATP, no NADPH. It just happens, which is great. But taking it from PGA to G3P, we see that you need both ATP and NADPH in that section. know if that's coming from the light reactions. And so then once you get that G3P, some of it gets off, and a lot of it's used to make sure that it's not RABP. Because if everything's going okay, the plant can keep its stomata open all day long, taking in CO2 all day long. So you have to have a good bank of RABP that it can react with all that CO2. Questions on the Calvin cycle? All right, well, the enzyme. We talked about that last week too. Rubisco, kind of a funky enzyme. It evolved a couple billion years ago when there was no oxygen on Earth. So it never had to worry, so to speak, about oxygen. However, the last two letters in the acronym, the C and O, the C stands for carboxylase. So it's the ASE. So this Rubisco enzyme has a carboxylase function, but it also has a function. So I like to think of it as it's a sloppy enzyme. It could, excuse me, it could take in CO2 into its active site or oxygen. It's really not too good. And so it is a competitive reaction. Whatever's around Rubisco in higher levels, that's what's going to win. So if there's a lot of CO2, great. If there's for some reason not a lot of CO2 but enough oxygen, then oxygen comes into the active site of Rubisco. And so that's what kicks off the PCO cycle. Oxygen wins. Well, that can create some problems. So here what you're seeing is if RUBP reacts, geez, sorry. If RUBP reacts, you get one molecule of PGA and then a 2-carbon molecule, phosphoglycate. Calvin cycle doesn't deal with phosphoglycate. It just doesn't have any way to plug it in to the cycle. And so the plants have evolved this pretty complicated mechanism in order to deal with phosphoglycate and try to get something good out of it, which would be another PGA. If they can create a situation where they take that phosphoglycate and run some reactions and end up with PGA, that's good. PGA can get right into the Calvin cycle. All right. So here, you're looking at the flow chart that we looked at last time, where it just all depends. What is Rubisco seeing? More CO2 than O2 or more oxygen than CO2? And so off to the right, you see what can happen if there's plenty of CO2, plenty of CO2 coming in. That's great. You got the Calvin cycle cooking along. Off to the left is what can happen if oxygen wins and comes into the active site of Rubisco. So this rendition where they show 3 arrows, that's a whole lot of stuff happening. It's a lot of extra effort by the plant to convert that phosphoglycate into a molecule of PGA. And so here you're looking at it, so I can show to you that it's a 2-carbon phosphoglycate, and the Calvin cycle cannot deal with that. And so plants evolve this mechanism where so right in the very top left of both of these images is where you see the kickoff of the PCO cycle, where you see oxygen right there and RUBP reacting. And you have to imagine Rubisco sitting there. And when you do that, you get a molecule of PGA, which is great because that can feed right into the C3 cycle. But then the phosphoglycate, you can see on the left here in both images, that has to get sent through all of this in order to get converted to a PGA. So you start off in the chloroplast, and then the glycolate has to go and find the peroxisome to do the next phase, and then the glycine has to find the mitochondria to do the next phase, and then back again. It's a lot. There is extra energy needed to get it to completion. You can see here ATP is needed at the very end, so you expend extra energy. There are some other losses, including over here in the mitochondria. I do want you to know that in the mitochondria, we see carbon dioxide getting created. Instead of getting fixed into sugars, we see it getting created. That's not a good thing. That's not what the plant should be doing. And because it's over in the mitochondria, they consider it lost. It won't get fixed into the sugars. So they say it's a loss of carbon in the form of carbon dioxide. So that's why they often refer to the PCO cycle as photorespiration, because the plant's giving off CO2 and it's just lost. It's not getting used to make sugars. So that's a big deal. The expenditure of ATP, that's a big deal. And then just the complexity. It takes time and effort. And so if you take a plant in the lab and you force them to do the PCO cycle for long stretches of time, they don't do well. They don't grow very well. It takes them longer to get flower production, fruit production. And so they know it's a stress on the plant. So in general, most plant physiologists would say, yeah, you don't want to plant on a regular basis. And really, it all comes down to the fact that Rubisco is a sloppy enzyme. It's very egalitarian about whatever comes into its active site. It doesn't care. Carbon dioxide, oxygen, it's fine. It's all good. So it's never adapted that high binding just for CO2, that very specific binding. So it's a problem. So a lot of the plants that we see here in this region of the world are what we call C3 plants, where they do the Calvin cycle. But if they experience some adverse habitats, they could kick into the PCO cycle. So in order to kick into the PCO cycle, oxygen has to win and come into the active site. What kind of environmental states might the plant be experiencing where things would shift, where there's more oxygen instead of carbon dioxide available around it? What would a plant be doing to start limiting how much CO2 is coming in? What would they do? How does a plant stop CO2 coming in into their leaves? Yep, close the stomata. So if you're a plant and you're like, oh, I'm going to close my stomata, why would you make that decision? It has to be a pretty serious reason for that. Why would you do that? Drought, absolutely. So if your roots are going, oh, we're getting this dry soil here, and now there's even more dry soil. I only have some water in. I can't walk over to the refrigerator and pour water. I got to deal with it the only way I can to be rooted. I'm going to shut down. So if the tree plant in this area, think like mid-July, really hot day, no rain for weeks, they're going to be doing. They'll have their stomata closed for large stretches of the day. CO2 is not coming in. So now inside the leaf, the CO2 levels are dropping. Oxygen is still there though. So now you get that shift where there's more oxygen around the Rubisco. And so under drought conditions, the PCO cycle will kick in with the plants around here, the C3 plants. Now, if you think about certain parts of the world where drought can be an issue, and then also think about higher temps. Like we have pretty modest in general or some temperature. But if you go to the equator, you can have much longer summers, much hotter temperatures, and there are periods of drought. So those plants are even more dire conditions where if they keep closing their stomata, that's going to force them to run the PCO a lot. doing this thing a lot, not good. And so what we find is certain plants have evolved mechanisms. Plants that live in the, especially along the equator, in tropical, subtropical, they've evolved a pretty slick system called the C4 pathway. So the C4 pathway, or the C4 cycle, we call it. So what you're seeing here is this upper left version. This picture is of a leaf that's a C3 plant, like what we have around here. The lower right image is the cross section of a leaf that's a C4 plant. And if you start going back and forth and looking at these two images, you can see that there's a little difference in their design of how they have their cells inside the leaf with regard to the bundle sheath here, this ring of cells. We call it the bundle sheath cell. So you're going to need to know that anatomy. Okay, so that's a ring of cells around the vascular tissue. And you can see in this, the way the artist has plumped them up, they're much bigger compared to what you see over here. The C3 plants have a bundle sheath ring of cells too, but they're not as built up. They're not as big and plump. And there's far fewer chloroplasts in a C3 plant in the bundle sheaths. So big, enlarged bundle sheath cells. And then don't exactly like how the artist drew. They should have put some spongy mesophyll a little bit closer to the bundle sheath. So you'll see some pictures here in a minute. But what they're going to do now is they're going to take a bundle sheath cell and a mesophyll cell, and they're going to zoom into that on this next image. So when you look at a C4 pathway, what I've written here, this CO2 concentrating mechanism, basically that's what that cycle is designed to do. It's designed to create a situation where Rubisco is fed lots of carbon dioxide all day long. And if you can keep that happening where Rubisco is surrounded by lots of CO2, then you won't have these CO2. And so it concentrates CO2 around Rubisco, and that is a great thing. I also have here spatial separation. So we're going to see how certain things happen in physically different locations inside the leaf. So some parts will happen in a mesophyll cell of the leaf, the parenchyma cell, and then some will happen in the bundle sheath. So this, they're going to enlarge this now. And so off to the right, you're seeing a mesophyll cell, and then the darker green is the bundle sheath cell, okay? So they're showing that different things are happening in those two physical locations inside the leaf. So what we see happening here, when CO2 comes in, it goes to a mesophyll cell, parenchyma cell, and it will react with this molecule called PEP, P-E-P or PEP. So that's not RUBP, totally different molecule. All right, so CO2 reacts with PEP, and then there's a totally different enzyme. We have an enzyme called PEP carboxylase. That's not Rubisco. So when CO2 first comes inside the plant through the stomata, it won't even go right to Rubisco. Instead, it goes right into the active site of PEP or PEP carboxylase. That's a good thing. PEP carboxylase is a really picky enzyme. It's like, nope, I'm only taking CO2 into my active site. Oxygen, nope, not allowed. So it has, in the fancy, sciencey way to say it, we say it has a high binding. It just really loves to bind to CO2 into its active site, not oxygen. So this is great because it allows for very efficient binding of this PEP molecule to the CO2. And then you see the product of that is a 4-carbon molecule called oxaloacetate. Another way to say that is oxaloacetic acid. So often we abbreviate it OAA, oxaloacetic acetate. And so then you can see that OAA converts to this other acid that we call malate. So you can call malate or malic acid, same thing. Okay, so all of that's happening in the mesophyll cell. Then you can see at that stage, the malate physically moves over to the bundle sheath cell. So I like to think of malate as a vector. And when we talk about vectors in biology, it means it's something that's used to move from point A to point B. So malate is a vector moving carbon dioxide, which is part of its structure, from the mesophyll cell into the bundle sheath. So I like to think of malate, it has, CO2 as part of its body, so to speak. And so now malate is going to carry that from the mesophyll into the bundle sheath. And then once it gets into the bundle sheath, that's where Rubisco is. It's in the bundle sheath. Rubisco's not over in the mesophyll cell. They spatially separate what's going on here. So Rubisco is in the bundle sheath cell. Malate comes over and delivers CO2 right to Rubisco. And then that CO2 can react with RUBP, and you have the whole Calvin cycle cooking along here in the bundle sheath. Now, if malate delivers a CO2, cleaves that part of itself off and gives it to Rubisco, the other product will be pyruvate. So because this is a C4 cycle, it's a cycle, we got to go back now and make more PEP to keep this all going. And so that pyruvate will get moved back over, back to the mesophyll cell. You see that there is expenditure of ATP though, that's really important to pick up. Okay, ATP is getting used here. So there is a cost to doing this cycle, extra cost, extra ATP. But when you do that, you'll end up again with pyruvate, PEP. And then the cycle can keep going. So they're physically separating. Rubisco is tucked away and hidden in the bundle sheath cell. CO2, when it first comes in, is getting reacted with in the mesophyll cell with a very picky enzyme. So that PEP carboxylase is only going to take carbon dioxide in, react it with PEP, that gets converted to oxaloacetate, which gets converted to malate, and then malate is like, the vector. It's going to take that CO2 that's part of its body and shift it into the bundle sheath where Rubisco's sitting there. And so the malate just keeps feeding CO2 to Rubisco, which is great. So it really does concentrate CO2 around Rubisco in the bundle sheath. And then you just got to keep the cycle going. You have to convert the pyruvate back to phosphorylated pyruvate. So there is an extra ATP cost, but the benefit is worth it. The benefit outweighs the cost. And so plants that have evolved and adapted to the tropical area, the equator, subtropical area, a lot of those plants. And what they find, they look very carefully at, how does this help them? They can keep going under drought conditions much longer than if there's a nearby C3 plant that just does C3. These C4 plants will win dry, hot conditions where there's stretches of drought, but it's still really hot out. They win. They do better. They can have a more efficient photosynthesis rate, greater production of sugars, greater overall biomass. They'll complete their life cycle in a timely fashion. So they win in those types of habitats. C4 plants are really good. And they can even close down their stomata on C4 plants. If they're like, ooh, I'm getting into a drought, they can close down their stomata to like 10%, but still be super efficient at photosynthesis. Where a nearby C3 plant does that, their photosynthesis was 10%. They don't use the low. The most famous one is corn. Yeah, they do C4. And we grow corn up here. So they're kind of at a disadvantage to grow up here because they do that C4. They're expending ATP when they don't really need to, because up here they don't have long droughts like they do where they're native to. So in some ways, growing corn in this environment, it is not the best thing in the sense that the corn is expending extra ATP when they really don't need to. But they can't turn this off. They can't turn the C4 cycle off. So, okay, so with the C4 cycle, you're thinking about the spatial difference. Certain things happen in the mesophyll, certain things happen in the bundle sheath. Realizing that Rubisco is in the bundle sheath and won't see that carbon dioxide initially coming in. It's going to see the carbon dioxide coming off the sheath. And then if the cycle is continuing to happen over and over and over, that means there's lots of malate coming and delivering that CO2. And so that Rubisco is going to have lots of carbon dioxide. The other thing that's happening, probably we could show on the diagram, is that the light reactions of photosynthesis, they don't really happen in the bundle sheets, which is good because if a plant is doing a light reaction, oxygen, you don't want oxygen near Rubisco. So the plant, C4 plants even do that in separation. They say, nope, we're not going to do the light reactions in the bundle sheet. Because when I do light reactions, part of my products are oxygen. I don't want oxygen in Rubisco. I'm trying to hide Rubisco away from oxygen. So I'm going to stuff it in the bundle sheet. Don't do any light reactions. All right, and so again, just really good at creating a situation where Rubisco is getting fed lots of carbon dioxide and not seeing that oxygen. So C4 plants adapted this pathway and are quite efficient. Even with that extra ATP, they're more efficient than C3 plants. If you do a head-to-head test in a tropical setting, C4 plants definitely have the advantage. This is another version of it. I just put this in here in case this works better for somebody's brain. If you want to look at it this way, this is just another way, okay? All right, so this is in words or written, what I've been telling you verbally. So you have that carbon dioxide reacting with that PEP carboxylase enzyme, and then it binds to PEP, and there's the full spelling, phosphoenol pyruvate. You don't need to know the full. I tend to go with abbreviations. So if I ask you about this on the homework assignment or the exam, writing in PEP, PEP is just fine. You don't have to spell it all out, okay? All right, so once you have carbon dioxide coming in and reacting with PEP, with that PEP carboxylase enzyme, you end up with OAA. And that it's just really a good situation because that enzyme, PEP carboxylase, is super efficient. It only cares about CO2, does not care about oxygen. And so you won't have any issues there in terms of any photorespiration. So when you look at these C4 plants, they suppress very nicely the photorespiration process, the PCO cycle. That hardly ever happens in C4 plants. It's got to be really, really extreme conditions for them to kick into that. So C4 plants just pretty much have figured out a way through this pathway, through this cycle, to greatly reduce the PCO cycle. So that malic acid or the malate is just very handy. It's able to incorporate CO2 as part of its structure, and then it'll just move it over into the bundle sheath cell and deliver it to Rubisco sitting in the bundle sheath cell. And then the Calvin cycle can cook along in the bundle sheath. So the Calvin cycle really isn't happening in the mesophyll. They leave that for the bundle sheath cells to do. So they really are very efficient in tropical conditions. But if you think about C4 plant up here, it's not tropical up here. So if you put a C4 plant here, they actually do worse than a C3 because they are expending that extra ATP when they don't really need to. So it is a cost, but if they're in their native habitat, it's worth it because they're going to be experiencing drought along with high temps. When you look at C4 plants under the microscope, when you look at their leaves, So this is, I believe this was corn or maize. So they've done a cross section of a leaf, and so you can see how plump the bundle sheath, the ring of cells are, very, very plump. And then the mesophyll cells, the spongy mesophyll cells, are usually kind of hugging up to the bundle sheath because they have to pass molecules back and forth to do the C4 cycle. And so you tend to see that as well. Another thing to pick up here, this is a corn leaf. If anyone's picked corn in a cornfield, do you think, well, does anyone know, is corn a monocot or a dicot? Want to take a guess? It's a monocot, and you'll learn more about that as you'll learn how to identify. And well, I'll tell you right now, one way to identify a monocot is when you cut a leaf in cross section. As you see here, all the veins are coming out at you. They're all in the same orientation. So that means all three of these veins are parallel to each other. They're running parallel. That's a dead giveaway that you're a monocot. Monocots have parallel venation in their leaf. And we'll learn more about that as we get into leaf anatomy. These three, so those are the veins, and the bundle sheath is around each of the, so the xylem and phloem is inside there. So these veins, when you cut a monocot leaf in cross section, it's like my fingers are the veins. That's the image they're coming out at you, but they're running in parallel to each other. So the veins are parallel to each other, and that's a dead giveaway that you have a monocot leaf that you're looking at. But here, this artist drew it a little bit better. Up top here is a C4 leaf, and I like how they drew this better, where you see the mesophyll cells hugging up to the bluish bundle sheath cells. And that makes sense that they hug up to each other because you gotta have tunnels, plasmodesmata tunnels between the two, because you have malate, coming in, you have pyruvate leaving. So you gotta have those molecules constantly going back and forth so that you can keep doing the C4 cycle. So it makes sense that they have some good physical proximity, lots of those tunnels, those plasmodesmata going between them. So there's a handful of C4 plants. Corn is the most famous. Here you're seeing image of sugarcane. So hugely important in our food industry, both of those are. This is a field of sorghum, which also is actually gaining in the usage. That used to be used just to feed like cattle, but they're actually incorporating it now in a lot of human food directly. So 3 huge crops are all C4 plants. Okay. So C4 plants, on this image, what you're seeing here is that on the left here, if you give a C4 plant more light, so that's what you're seeing here, brighter and brighter light quality as you go off onto the Y. You give the plant more light, it does more photosynthesis. C4 plant, more light, more photosynthesis. More light, more photosynthesis. You can see it going up, 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 up. And it goes up much further than a C3 plant. The C3 plant has limitations. It's like, I can't do anymore. I'm going as fast as I can. C4 plants, because of that C4 cycle, they're way efficient. Temperature, you're seeing here, hot temperatures, C4 plants win. They're going to do better photosynthesis than a C3 plant. But here, this is interesting. The far right, think about global warming. We have CO2 levels going up. So that's what they're showing here, CO2 levels going up as you work your way to the right of the x-axis. You can see that C4 plants, they slow down, but C3 plants keep going up. So what that's saying is with global warming, C4 plants are already so efficient at photosynthesis that it doesn't matter. Feed them more CO2 and they're like, yeah, I'm already super efficient. But with C3 plants, you feed them more CO2 and they like it. They're like, ooh, good, I don't have to run the PCO cycle. And so they can actually do better. Thinking about global warming, C3 plants might actually like it for a while anyways, right? So we'll stop there and we'll review C4 on Thursday, and then we got to cover one more pathway.